our technical chief here. Several years ago, when our lab decided what we really needed to know for our Parthenone work was what genes normal 8-cell embryos express. And in trying to understand how to do that, rather than simply do a bioinformatics study where we put out all the genes and Venn diagrams and everything, we decided it might be more instructive for what we needed to know to go through it a little more piecemeal. And so we looked at some cell cycle genes because I had it in my head that the problem, because I do have a background in DNA replication, I decided that that was the problem. That the reason human parthenones didn't do well is there was some kind of error in DNA replication, and that we needed to see what early embryo DNA replication was normal. So we looked at DNA replication genes. And as a part of that, one of the first things that popped out of our microarray set was the enormously high expression of the circadian rhythm genes. Now, we were comparing the 8-cell embryo to embryonic stem cells and to Yamanaka's fibroblasts before and after induced pluripotency. And the circadian rhythm genes in those cell lines was flat, was not on. So what on earth, what kind of a circadian signal can an early embryo even respond to? Why were those genes so active? Was it just a holdover from the egg? They've only been in culture 3 days. And so we got and I began to realize I know how to spell circadian rhythm. I don't know anything about them. And I found a book called Clocks and Rhythms that came out of a Cold Spring Harbor Symposium at some point. And I had actually been invited to a Red Sox game. And I spent the Red Sox game reading Clocks and Rhythms. And that's how we became we reached out to Dr. Davis, who's with us today, Fred Davis from Northeastern, and said, we don't know anything about this. This doesn't make any sense. Help us. So then we got involved in doing some circadian rhythm studies with a transgenic mouse called Per2Luck that was developed by Joe Takahashi, who spoke with us last time. And so we're still trying to understand the role of circadian rhythms, and I won't go any longer, but we have an expert with us today from UMass Amherst, Eric Bittman, who has done some interesting work looking at circadian rhythms and reproduction in general. And so I'm delighted. Dr. Bittman started his work, his life, his science life at the University of Pennsylvania. He then went to UC Berkeley for a postdoc. He then went to Michigan. In his postdoc, he was really interested in endocrinology and psychiatry and things like that. And then he went, you can correct me if I'm doing this wrong. And then he went to Michigan, and then he spent some time at Rockefeller, and he's been at the UMass in Amherst since 1985. He now is emeritus, which means he doesn't have to teach anymore. And he still has an active lab doing his circadian rhythm. So welcome with me, Dr. Bittman.
Thank you, Ann, and thank you for the opportunity to speak today. My talk will start out with sort of a primer on circadian rhythms and tell you a little bit about the cellular and molecular basis of rhythms, and then broaden out to more of a physiological perspective, talking about how the circadian clock is entrained by environmental cues, and get into also how the circadian clock is used to measure the length of the day and restrict reproduction to appropriate times of year. And I'll, in the first part of the talk, get into some of the physiological importance in terms of the temporal coordination of physiology and how the circadian clock keeps the trains running at their appropriate times, which is not necessarily synchronous, but to ensure that compatible events and the substrates peak at the same time, and incompatible events might be oppositely scheduled. Then I'll get into reproduction. To be frank, I'm a neuroendocrinologist, So my interests and work has been a bit upstairs from the subject of talks of the other speakers, but there is an important role of circadian clocks all the way from the brain down to the uterus that I'll talk a little bit about. A lot of our work is guided by or informed by effects of clock mutations, which affect ovarian cycles, especially the LH surge, but also subsequent events. And I'll end up talking a little bit more about seasonality and the role of circadian rhythms in annual rhythms. So it's a bit of a challenge, optically as well as technically, because I can't see the cursor here. Okay. So the fundamental experiment and observation that circadian biologists do is to, of course, observe rhythms in living animals. And it turns out that just looking at sleep-wake cycles, activity is a handy set of hands of the clock. And when we put an animal like a mouse or a hamster in a cage with outlet access to a running wheel and have it on a light-dark cycle, a period of 24 hours, they will, of course, adopt the periodicity of the environment. And one might be excused for just assuming that the animal is just passively responding to these periodic environmental cues. But a critical experiment is to transfer the animals into constant condition. And here we've turned the lights out and we observe that rhythmicity in behavior persists. The period of rhythm, for example, the interval between the onset of activity on successive cycles, however, is no longer exactly that of the environment. It's no longer a 24-hour period. But in this case, with this mouse, it's running a little bit earlier on successive days. So we conclude that the rhythm can free run, that's our standard terminology, with a period that differs slightly from 24 hours, and that's where the term circadian or circadian comes from. And as I will show you, these observations with respect to behavior are actually a convenient set of hands of the clock and really reflects what's going on physiologically with respect to hormone secretion and other physiological events. So our conclusion is there's an endogenous oscillator, or maybe more than one, that's generating rhythmicity, but that the clock, the circadian clock, can be entrained by environmental cues. And in order to identify the mechanisms down to the cellular level, it's been useful to take advantage of mutations to identify specific genes that either alter the period of the free-running clock or in some cases actually can eliminate rhythmicity altogether. So I've been working with two different mutations, specifically in hamsters. And why hamsters? We'll get to this later in the talk. Hamsters are very sensitive to the length of the day, and to study seasonality of reproduction, you really want to work with a hamster or maybe a sheep. Lab mice are really not so good, especially not the C57 strain that's so popular in the circadian field. But here are representative records from wild-type animals, and you can see on the 24-hour time base over successive days, the wild-type animal has a free-running rhythm whose period in constant darkness is about 24 hours, actually, in this animal a little bit longer. And starting in about 1987, the first period mutant was discovered in Mike Menaker's lab, which was deemed the tau mutation. This is not related to the tau gene. Tau is the Greek letter we use to reflect the period of the rhythm. And we see in an animal that's heterozygous for this mutation, the clock runs fast. The period is about actually 22 hours. It's running about 2 hours earlier every day. And in tau homozygote, the period's actually 20 hours. In other words, this is the dominant negative mutation. And it led us to define the tau homozygote as the super short hamster. And we were working with tau homozygotes in our lab. We were doing transplantation experiments, which I'll describe briefly in a minute. And on this tau homozygote background, we got a second mutation on the super short, which we called super duper, because the period actually was no longer just 20 hours, it actually shortened further to 18 hours. So we suspected and then later confirmed that indeed there was a second mutation on the tau background, which is of course just duper. And when we got it on a wild type background, the period is about 23 hours, just a little bit longer, but in the same range as the tau heterozygote. So here are group data showing the circadian periods of wild type, duper, tau heterozygous, tau homozygote, super shorts, and the super dupers. So the availability of these mutations made it possible to tease apart the basis of the circadian clock and the discovery that indeed there is a cell autonomous clock represented here, which actually works on the basis of transcriptional translational feedback loops. The heterodimer proteins B-mel and clock is kind of at the center of the story, and B-mel clock is a transcriptional activator which works upon binding to EBOX motifs in the promoters of a number of important genes, some of which are shown here. And we especially emphasize the period genes, especially PRO1 and 2, and the cryptochromes, CRY 1 and 2, whose transcription and then translation are ultimately activated by B-mel clock at specific times of day. PER and CRY, upon translation, undergo important post-translational processing, especially by casein kinases, and phosphorylation can regulate translocation to the nucleus, where they actually inhibit their own transactivation. So here's our basic negative feedback loop. The period of the rhythm is also affected by the degradation of PER and CRY by specific enzymes. There's another loop here, some people regard it as ancillary, but it's important, where by the retinogorphin receptors, ROR and REVER will act on retinogorphin elements to regulate B-mel cyclicity. So also indicated on this slide, and perhaps deserving more evidence, is this little box here, which are clock control genes. So we distinguish between the core clock genes, such as PER and CRY, and perhaps ROR and REVER. Those are genes that are regulated by B-mel and clock through the EBOX, which, if mutated or if knocked out, will result in changes of period or lack of rhythmicity. So we refer to them as in the core of the clock. Here, the CCG refers to clock control genes, which are kind of the working class heroes of physiology, in that these are genes distributed in every organ that might be directly regulated by EBOXes and that are rhythmic. And there are thousands of genes, as actually Joe Takahashi described, if any of you were here in 2019 and heard his elegant talk, that the whole transcriptome is orchestrated by the circadian clock, because these clock control genes are controlling many metabolic pathways. It's not that every enzyme is clock controlled, but in just about every organ, it's often the rate limiting enzyme in every pathway has circadian control through a mechanism like this. So while discuss the results that we've gotten looking at the effects of B-mel deletion, which are often most severe because B-mel is really at the core of the clock. And in fact, B-mel is the one gene, if you eliminate B-mel and B-mel only, you lose all circadian rhythms. And then we've been doing work following up on the tau mutation, which is actually a gain of function of casein kinase 1 epsilon, which shortens the half-life of HER2 and speeds up the clock. And most recently in my lab, we discovered a new mutation, and I just talked about that, which is DUPR, and through next generation sequencing, in collaboration with John Hopkins, we've identified DUPR mutation as a single base deletion in exon 4 of PRI1, which leads to lack of expression of cryptochrome 1. So a lot of this work has been facilitated by having a luciferase reporter such that we can observe either in dispersed cell preparations or in fibroblast cultures often, the clock running in real time. So here's an example of what we can observe in time lapse in a cell culture where these are actually cells from an area of the brain I'll talk about shortly. Each of these cells has a HER2 loop fusion protein and is cultured in the presence of the luciferin substrate. And all of the cells are actually oscillating with a 24-hour basis. And this graph is showing this individual cell shown in the box, and it goes through 2 weeks of perfectly regular cycles, but it's out of phase with the other cells in a dispersed cell culture. So whether we're looking at neurons or fibroblasts or hepatocytes, we will see these rhythms. In the brain, the synaptic coupling will ensure phase coherence of the individual cells. Not so clear that that's the case, and probably isn't the case in other tissues, and it's the role of the brain, and specifically a pacemaker in the hypothalamus, to determine the circadian phase and to maintain synchrony within the peripheral organs of their dispersed individual circadian oscillators in their cells. So as reproductive biologists, perhaps it shouldn't surprise us that the locus of this kind of control is in the hypothalamus. And indeed, the suprachiasmatic nucleus of the hypothalamus is critically important. Lesions of this structure make animals arrhythmic. And here's an experiment which we replicated, originally done by Martin Mouth in Medical's lab, showing this is on a double plotted activity record. So we're showing 48 hours. On the left-hand side, the computer has placed a dot to indicate where it thinks by its algorithm that activity has started for that particular cycle. And we see in this super short host that the period is very short. It's running with a period of about 20 hours. And at this point, we lesion the suprachiasmatic nucleus, and the animal loses its circadian rhythms of locomotor behavior. I will say parenthetically, it also loses all the other physiological, including endocrine rhythms that we can measure. And using a technique that we devised in the mid-80s, we found that we were able to transplant the SCN from fetuses into the lesion cavity. And in most of the animals, the circadian rhythms come back. But the trick and the wrinkle of that graft experiment that was first done here made use of the fact that you can transplant into a super short host a wild type SCN. And you see when rhythmicity comes back, look at the period of the rhythm. The period is out of the donut. Whereas all the other cells in the body think the clock should be going this fast. Just transplantation of the SCN is enough to make at least the activity rhythms adopt the period of the SCN. So this is the critical evidence that the SCN is not just permissive, but it's actually the master pacemaker that coordinates circadian rhythms throughout the organ. And making use of the luminescent reporter, the HER2 loop preparation, we can actually work watching a time lapse in a slice of hypothalamus. Here's the third ventricle, here's the optic chiasm, here's the suprachiasmatic nucleus. We're looking at the HER2 driven luciferase. that is oscillating cycle after cycle. So we have a system in which there's a master pacemaker in the brain, and then there are peripheral oscillators distributed throughout the body. And the importance here is that the time givers, site givers from the environment are going to be the cues that allow the endogenous rhythm to entrain to typically 24-hour period of the environment. And of these site givers, it's the light cycle that's the most powerful. And the SCN is not just coincidentally placed above the optic chiasm, it actually receives a privileged retinal production that's responsible for entrainment, a very interesting story I don't really have time to go into. The brain's pacemaker is controlling the peripheral clocks, which also have, of course, their own cellular oscillators that don't maintain phase coherence very well, but via endocrine and autonomic signals principally, and perhaps through behavior and regulation, body temperature. The brain's clock is ensuring 24-hour coherence of the peripheral clocks. The significance of this also is that we know that, or have found over the recent years, that if we disrupt the normal temporal relationships, in other words, if we induce misalignment of phase between or perhaps even within the cellular clocks in various organs, we get pathologies or we aggravate existing pathologies. So we see this in our modern life. Many people who travel across time zones and experience jet lag, or people who work on various shift cycles, and perhaps even people who change their social schedule. You know, on the weekend they stay up late and then they pay the price on Monday. Part of the problem with that is that they're experiencing a misalignment, a temporal misalignment between and among circadian clocks. So, as I said, I'm very interested in hamsters, partly because I'm interested in seasonal reproduction. And in collaboration with Zonda Wang at Utah State, we now have a knock-in HER2 loop hamster, and we've documented that not only the SCN, but also in many peripheral tissues, including reproductive tissues, the pituitary, the ovary, and the uterus might be of particular interest to this group, but also the other organs that we looked at have circadian rhythms when cultured. And in collaboration with Jen Evans at Marquette University, we've been looking at brain slices. Here's pituitary, comparing the oscillations in cultured pituitary. Black is the wild type, the blue is the duper mutant, and the green is a tau heterozygote. And this documents what I was saying about the activity being a nice set of hands of the clock, because we see the same kind of pattern of the effects of the mutations when we look at the peripheral organs and the luciferase reporter rhythms. And we find that the suprachiasmatic nucleus of the duper hamsters has a shorter period, as shown here, and it also damps out after fewer cycles than does the wild type, so that the duper mutation, which eliminates the expression of Cry1, changes not only the cell autonomous oscillator, we think it also changes the coupling between cells within the pacemaker. Okay, so all that by way of introducing circadian rhythms and explaining their basis. Let's talk about reproduction. So it turns out that all sorts of cells up and down the reproductive axis are expressing circadian clocks. My interest has been principally in what is it that controls pre-ovulatory LH surge. And this diagram kind of emphasizes some of the loci, but also clues you into the idea, and Joe talked about this in the 2019 symposium, that in the ovary as well as in the uterus, we also have circadian clock function. But within the brain, the SCN is critical to ovulation. And in fact, in order to generate an LH surge, you of course need the release of GnRH, gonadotropin And there are two conditions required for that in the animals we study, mice, rats, and hamsters. First of all, we need high levels of estradiol, as occurs in the late follicular phase, but that's not enough. We also need an intact circadian clock, because if the SCN is lesioned, the animal will never ovulate again. Interestingly, if we do a transplant of the SCN that reinstates locomotor rhythms, it doesn't bring back the ovulatory rhythm or the LH surge. And we think that the signals from the SCN that control ovulation are neuronal projections, whereas the outputs of the SCN that control locomotor behavior and have some of the other rhythms are diffused signals. Some of the neuronal projections of the SCN that we think are important are shown here. My laser pointer a little bit on the blank. The SCN has many important projections shown here in this diagram. We see the SCN is a pretty complicated structure. The ventral part of the SCN, which actually receives the retinal projection, is dominated by cells that make vasoactive intestinal polypeptide. That's important for coupling of the oscillators within the SCN. The dorsal SCN makes vasopressin, a parvocellular small neuron population, and the vasopressin cells have very important projections to a group of cells that make an important peptide called kispeptin, which has been very exciting because in about the last 10 or 15 years, because it turns out that the AVPV, the anteroventral periventricular nucleus kispeptin population, project directly to the generation neurons that is necessary for the LH surge to occur. So it's possible that the most important link here is vasopressin to AVPV kispeptin to GnRH. However, there are projections, some of which contain VIP from the SN directly to the GnRH cells. There's a population of cells in the dorsal-medial hypothalamus that makes a femurfamide peptide, some call it gonadotropin inhibitory hormone, that projects to the GnRH cells and also regulate it, and may also directly control the pituitary. So all of these are perhaps important to varying degrees in controlling the LH surge. And the circadian clock in the ovary, there's some evidence from Mike Selleck's, also in his lab, suggesting that a clock in the ovary controls sensitivity to LH in the ovarian follicle. So even further down in the axis, and perhaps of greatest interest to this group, are looking at effects of the circadian clock on implantation. And in early days, what people were doing, early meaning like, when was this? Early 2000s. It's funny how the early 2000s got to be the early days so soon. People were doing global deletion. So you're knocking out female all the cells of the organism, and that causes multiple defects, because as I've said, there are circadian clocks in every organ controlling all kinds of functions. And then moving on, it became possible to do conditional deletion using Cree-Lox approaches to allow the identification of the role of circadian clock in specific cell types that influence reproduction and other functions. And Chris Bradfield's lab, they engineered mice in which BMAL was deleted specifically in cells that make steroidogenic factor 1, important for production of all the steroid hormones. And this resulted in the failure of implantation. So here we have control the female floxed animals without the SF1 Cree, and they are able to have normal implantation. But among the animals with conditional deletion of BMAL, specifically in the steroidogenic factor producing cells, with corn oil, they don't, of course, implant. So there's a failure of implantation if female is knocked out in steroid-producing cells. However, if the animals are given progesterone, we get normal implantation, and to a somewhat lesser extent, if the animals are given exogenous prolactin, it can result in normal implantation. So here's an example where female is important in reproduction at the level of implantation subsequent to the early stage. My work has utilized a similar approach. As I said, global deletion of female is devastating and eliminates the LH surge in over intact mice. Here's work from Chu in Florian Storch's lab, showing in a wild type oncoestrus around the time of lights out, you get in each of these individual mice, except for one laggard, a nice coordinated LH surge, but with a global knockout of female. The animals don't surge, enough the difference in the scale of the y-axis. So clearly female is important, and circadian clocks are necessary. And we already knew that going back to the real old days, like the 50s, the work of Everett and Sawyer began to show that the circadian clock actually controls the LH surge, as I've been saying. And in fact, if you overoptimize a mouse or a rat or a hamster, of course, you won't get LH surges. If you implant them with estradiol, you will get a surge every day at the same time of day. So there's a circadian clock controlling the LH surge, as I showed in the earlier schematic. But our question was, what are the relevant cell types? Which cell types in the neuroendocrine circuit controlling ovulation must a circadian clock function in order to get a surge? And my student, Ajay Kumar, worked on this in his thesis. And as I mentioned, the SCN has different cell types. There are vasopressin cells that project to this important Kispeptin population. There are VIP containing cells that we think project directly to the GnRH. And of course, the GnRH cells are important in inducing directly the release of LH in the surge that causes ovulation. And certainly the GnRH cells, as well as the Kispeptin cells, as well as the vasopressin and VIP cells, all have circadian clocks from it. So what we did is we made transgenic strains. We made or took advantage of Cre recombinase being driven by vasopressin, VIP Kispeptin, or GnRH, crossed them with animals with the female flux construct. And what we found is that actually we could interfere with the LH surge by compromising the circadian clock in any of a number of these cell types that are all within the circuit. So the controls are shown here. The overoptimized estradiol implanted animals all surge around the time of lights out. But if we knock out female in the vasopressin cells, or in the Kispeptin cells, or in the GnRH cells, all of these result in either abnormally timed or absence of LH surges. We also have been doing work on the anatomy of the system, and it turns out that when we stain for GnRH, we can, of course, observe the important GnRH cells, mostly in the preoptic area and anterior hypothalamus through immunocytochemistry. We do double and triple label, and we confirm that the GnRH cells express VMAL and PER1 in antiphase. And then we noticed when we looked at the suprachiasmatic nucleus, these are GnRH axonal fibers in kind of the yellowish green. They go right through the SCN. So in a sense, not only does it seem that the SCN, the master pacemaker, is upstream from the GnRH cells, but maybe it's also downstream. And maybe it's regulating axonal production of the GnRH fibers, or at least the SCN is getting some handshake confirmation that the SCN surge is underway. As the GnRH pathways are passing through the coronal plane on the way to the median eminence to release GnRH and trigger the surge. Actually, the mouse, whoops, I shouldn't have done that. The mouse actually has it's less dramatic population of gene arranged fibers. I'm not going to try to go backwards because it might not work. So the other mutant, as I was saying, we've been working with tau mutant hamsters and with duper mutants. These both have fast clocks, and both of these mutations change the way in which the animal entrains to the light-dark cycle. Wild-type animals tend to wait to start their activity right about lights out. Dupers, as well as tau mutants, actually anticipate the onset of darkness. And there's reasons for this I don't have time to go into. But interestingly, that correlated with this change in the phase angle, the train phase angle, in the fast clock of mutants, we see the timing of the LH surges change. So shown here is the wild-type LH surge in ovary intact animals occurring around Zeitgeber time 9 or so, 3 hours before lights out. And in the duper mutants, it occurs about 3 or 4 hours earlier than in the wild type. So great interest in circadian rhythms, as I was saying earlier, stems from findings of the problems that arise from misalignment of circadian phase and the effects of jet lag and shift work, and the idea that loss of temporal coordination affects health. And what we've observed with the duper mutant is actually we get about a 4 or 5-fold reduction in the duration of jet lag in the duper mutant. So here are experiments in which we've taken hamsters. Here's a wild type, and we abruptly give it an 8-hour phase advance. And it takes about 2 weeks for the animal to re-entrain. It's only able to shift its clock by about an hour or 2 a day, and finally aligns normally to the light-dark cycle. When we shift it in the other direction with the phase delay, again, it takes about 2 weeks, shown here for this representative animal. Here's the group mean data. When we look at the duper animals, it takes them only about 3 or 4 days to re-entrain after either an 8-hour phase advance or an 8-hour phase delay. And when we look at the tau heterozygote mutants, we see the same kind of thing. When we do a phase advance, it takes about 5 days as opposed to the wild type, 2 weeks, to re-entrain to the shifted light-dark cycle. Interestingly, for all of the tau heterozygotes and for many of the duper mutants, when we give them a phase delay, they'll re-entrain, but they re-entrain antidromically. They decide to go forward by 16 hours instead of going back 8 hours, which we have some ideas about why that happens. But one thing that we wonder, as I've been arguing to you, is how well do the activity rhythms really represent what's going on physiologically? And in collaboration with John Hognish, who helped us identify the duper mutation as a Cry1 null, we've been doing RNA-seq experiments. So in these experiments, we take wild type or duper mutant hamsters, we entrain them to 14-10 light-dark cycle, and then we do acute phase advances, that I showed in the last slide, an 8-hour phase advance. And then we collected organs from groups of 5 animals on the 3rd day after the shift, and we had collected at 2-hour intervals over the 3rd cycle and performed RNA-seq. So far, our data are complete only for the liver. And what we're going to do, the next slide, is compare the phase of transcript abundance of various transcripts of various genes, the wild type versus the duper, first in the unshifted animals, and then perhaps more interestingly, on the shifted animals to evaluate whether the duper mutation, the absence of Cry1, changes the reentrainment of gene expression in various organisms, starting with the liver. And in this slide, here are the results. So the solid lines represent the unshifted population. So if we look at 4 genes of interest, so here's reverb, which is in the core circadian loop. And what we see is that the wild type, the transcript peaks around zygote time 10, just before lights out. And in the dupers, phase is advanced. And then most interestingly, when we shift animals and we look on the third subsequent cycle, that's shown by the dashed lines. So the black dashed line is for the wild types. They've only begun to get back into their normal steady state phase. But in the dupers, they've already achieved phase appropriate for that light dark cycle. Deomin binding protein is an interesting gene. It's in charge of xenobiotic resistance. So, for example, the length of time that the animal will stay out after you give it a barbiturate injection is controlled by DBP. So, the phase of DBP is shown in the wild types by the black and the unshifted animals, and in the dupers by the red solid line. When we shift them on the third day, the wild type has only begun to achieve this normal steady state change, but the dupers are already there. ALAS1, aminolevulinic acid, this controls heme synthesis in the liver. And same story. So there's an advance of the phase as there is for locomotor activity. In the unshifted dupers relative to the black solid line, the wild types, after we shifted them and we wait 3 days, the ALAS1 levels of the wild type are only starting to come back into the normal phase. The crime is already there. So the crime mutations seem to have greatly reduced misalignment and accelerated re-entrainment and antagonized or prevented jet lag. Interestingly, Cry2, which is also an important repressor within the translational feedback loop, is affected by the Cry1 deficiency, but also shifts slowly in the wild type and not so in the duper. So for the LH surge, we were interested in that women can experience amenorrhea or abnormal cycles when they experience jet lag. We replicated that in hamsters. We shifted animals on proestrus and then took serial samples on the next scheduled proestrus. And what we see is that the normal timing of the LH surge has been achieved in the duper mutants, but in the wild types, the LH surge is still a long way from getting to its normal phasing within 4 days after a phase advance. Okay, so to finish up, I'm probably stretching the time a bit. Sorry. Another consequence of circadian organization is coordinate reproduction seasonally. And humans are seasonal in their reproduction, and I can cite some work from Ronenberg and Ashef looking at historical records before society was so heavily influenced by electric lighting, showing that there are seasonal differences in fertility, and even today, initiation of pregnancy varies by time of year. This effect is much more dramatic in the animals we work with, and this is part of the reason why I choose hamsters, or this particular slide comes from Randy Nelson and illustrates the prognosis. These animals have a 3-week gestation period, and their offspring are born during long days, and as the days shorten, they cease having estrous cycles, and the testis will regress as much as tenfold in weight. This response is blocked by removal of the pineal gland, and it's controlled by the secretion of melatonin. And the production of melatonin is determined via sympathetic pathways, ultimately by the suprachiasmatic nucleus. And that's illustrated here. So the retinal production of the SCN hooks up via the PVN, the paraventricular nucleus, to autonomic control, and will result in production of melatonin that encodes the length of the day. So on the winter, there's a long duration of melatonin. and in the summer, the short duration of melatonin that is decoded by the brain to change reproduction seasonally. And there are two basic effects. One is that in a longer breeder, such as the hamster, the stimulation of reproduction is correlated with a decrease in the negative feedback potency of gonadal steroids. There is an experiment we did a long time ago where we took out the testes and implanted hamsters that were either on long days shown by the white bars or short days shown by the black bars. And we gave them varying scholastic capsule lengths of testosterone, which resulted in comparable levels of circulating testosterone and long or short day hamsters. But what we observed is that the short day hamsters, LH secretion is completely suppressed by a lower dose of testosterone than in long day hamsters. So the negative feedback loop is being modulated, and we found out this is by way of melatonin duration. So that's a steroid-dependent effect of day length on the negative feedback response. But there's also a steroid-independent effect in that if we remove the gonads and we look at the pulsatile secretion of LH, and this is an experiment we did in sheep, which are short day breeders, we observe these beautiful LH pulses. This is over the course of 6 hours. This is an ultradian rhythm, not a circadian rhythm. And what we see is that in anestrus and inhibitory days, we get these beautiful long period pulses of LH. And when we transfer the animals to a stimulatory photoperiod, even in the middle of summer, we give them short days. The LH pulse generator, which is now we know located in the kisspeptin cells of the hypothalamus, the pulse generator speeds up markedly under the influence of the pineal because of melatonin. If we take out the pineal, the change in photoperiod has no effect on the pulse generator. So again, the question is, where are these effects localized? And a lot of attention has now been drawn to an area of the pituitary gland. We're all familiar with the pars distalis. We think of it in terms of the gonadotropes releasing LH and FSH to regulate ovulation and growth of the follicle and maintenance of pregnancy. Of course, we also have the thyrotropes that release thyroid stimulating hormone to control the thyroid gland. But there's another area of the pituitary, the pars tuberalis, which is a cuff of tissue which also has thyrotropes. It surrounds the infundibular stalk, and it also has thyrotropes, but the TSH, the thyroid stimulating hormone, is actually not regulating the thyroid gland. Instead, it's regulating tenocytes that line the ependema of the third ventricle and regulate DNA methyltransferase, which affects or determines whether or not thyroxine is activated or inactivated, causing a remodeling of the brain. And these cells, these pars tuberalis TSH cells, are regulated by melatonin. And what's going on here is that when the lights come on during the day in the pars tuberalis, PER goes up, but when melatonin goes up at night, cry goes up. And the phase relationship, the interval between the peak of PER and the peak of cry, is controlled by melatonin, and specifically by the duration of melatonin, encoding the day length. And these changes in the negative feedback sensitivity of testosterone or estradiol in females, and probably the activity of the kisspeptin cells, ultimately being regulated by this remodeling in the brain that is mediated by melatonin acting on the pars tuberalis via TSH. So we got really interested when we discovered that our duper animals don't have cry. Hamsters, of course, are acutely sensitive to photoperiod, so we did an experiment where we released either wild-type animals from a light-dark cycle into constant darkness for 8 weeks, or did the same with dupers. And of course, we see with dupers that their clock runs very fast. And what we find is that the wild types The males regressed their gonads, their testes shrank by fourfold. The females stopped ovulating, the uterus weight declined in half. But the duper mutants, which don't have cry, don't respond. They don't shut off reproduction. So the cry deletion is also affecting seasonal reproduction as well as the LH surge and ester cycles. Okay, so to summarize, circadian rhythms are generated by transcriptional translation feedback loops. The SCN is a master pacemakers, influencing oscillators in the periphery. The alignment of circadian phase within and between organs is critical to normal function. I didn't have time to talk about other work that we've done with Duper, where their resistance to jet lag, their rapid re-entrainment actually ameliorates the effects of repeated phase shifts on heart disease that survive longer in the face of jet lag if they're cardiomyopathic. And also the Dupers have changes they have reduced effects of jet lag on neurogenesis in the hippocampus, which is another story. Circadian clocks regulate female reproduction, we think at multiple levels. And then finally, fertility varies seasonally, and circadian oscillators are used to measure photoperiod, and Cry1 seems to be necessary for that, so that mutations of core clock genes, including Cry1, alter both ovulatory cycles and seasonal reproduction. Thanks for your attention.
Exactly, two questions. Jose and David, and then the schedule for one I want to do.
Very tough.
That was the best explanation of the LH surge I've ever heard.
Very tough. What happens is we have tissue member in vitro that is not exposed to light at all. Why does tissue still have clock speeds ticking?
Can you just repeat the question, sorry.
So the question is whether isolated tissues, why do they still have? Okay.
They're not exposed to light.
Right. Well, you've got to have a photoreceptor. So some of you might remember David Whitmer's talk from 2019. He was working with zebrafish, in which case all the peripheral tissues are directly light affected. In mammals, it seems like the entrainment pathway must go by way of the retina. So the SCN is taking the light signal and ensuring that the peripheral oscillators are properly phased with respect to the external environment with respect to light. However, there is evidence that there are other cues, specifically nutrition cues. So for example, if you take a mouse and make it ingest all its calories in the middle of the day, which it doesn't want to do, the peripheral organs are responsive to foodborne signals, either maybe just glucose or by way of, say, pancreatic hormones, that kind of thing. So, okay.
That slide that showed the axon versus the super treating, is there any look using transmission electron microscopy to see if there's axonic synapses? And if so, are there cell bodies in the SCN? It's that those LH neurons are also deeply involved in reproductive behavior. And anybody who's worked with somebody who's bipolar or schizophrenia, where there's it's a fascinating in case of schizoaffective or bipolar hypersexual behavior, but more interestingly, there's almost a complete obliteration of any circadian entrainment of that. People go on a binge. So it'll be interesting to see, A, does it exist in humans, B, are there axons, and C, are they the glutamate, which is sort of the center of many of the leading theories now, schizophrenia, some sort of global glutamate toxicity.
So there are 3 or 4 questions.
Yeah.
I can't repeat all that question. So the part that I can answer, I can sort of answer, to answer, is whether there are axonal contacts with SCN cells. And yes, we see close appositions of GnRH fibers to both vasopressin and VIP cells, but as you intimated, you would need electron microscopy to identify that there are synapses. Alan Herbison, who I regard as the world's expert on GnRH cells, talks about dendrons, so that there is communication between GnRH processes and other cells. GnRH, there is evidence that projections of GnRH cells to the midbrain are important in reproductive behavior in rodents. We haven't really looked at that, and certainly haven't put that in the context of rhythms of sexual behavior, but it's a really interesting speculation. That's what I can say.
Dr. Bittman has been with us for the day, right? I know for the day, so there will be ample time to do about that. In the interest of now a little bit of time.